Yes, thanks, Irina, for the dynamic talk. Just a short housekeeping remark. As you see, we are going a little bit over time. I don't think this was too surprising with the interesting topics we are listening to here. So since we have the panel discussion afterwards, what we decided to do is obviously to give Michael the opportunity to also present for 15 minutes and maybe a minute more if needed. Then we will have a 5-minute bio break, very short one, so get prepared for that. And then we will start with the panel discussion, which will, however, stop at 4:30 sharp to give the after the sessions afterwards proper time to be addressed and get all the talks presented properly. So it's my pleasure now to introduce the last speaker of the session. Last but not least, it's Dr. Michael Hayden, the founder and CEO of Prelania, the sponsor of the PROOF-HD study. He has been working on Huntington's disease for, I don't believe it, 40 years, serves as a Killam professor at UBC in Vancouver and has mentored over 100 graduate students and postdocs and been the author of more than 900 peer-reviewed publications. So Dr. Hayden is committed to understanding the pathogenesis of Huntington's disease and translating that into products and services for patients and families. He led the development and approval of one of the 2 drugs currently approved by the FDA for Huntington's disease and will now update us on the status of the phase 3 clinical trial of Prodopedin, the PROOF-HD study. Go ahead, Michael.
Thank you, Ralph, and thank you to EHDN for giving me this opportunity to give you an update on the PROOF study. And the PROOF study has PR for Prodopedin, assessing the outcome of function in Huntington's disease. And today I want to give you an update on the progress in the study to its goals. Now, Prodopedin is an investigational drug. It's in clinical trials right now for HD and ALS. It's given orally twice a day, once in the morning and once in the evening. It's safe and tolerable. In prior studies, extensive studies, has a side effect profile similar to placebo. We now understand its biology, and the biology has driven the clinical design for the phase 3 study. It binds and is actually a very potent agonist for the sigma-1 and is highly specific at therapeutically active doses. This drug has shown maintenance of total functional capacity, particularly driven by patients with early HD. And certainly in open label studies, the effect has been shown to be durable for up to 5 years. Now, the big understanding of Prodopedin and its mechanisms of action came from human PET studies as well as in vitro studies, but these were perhaps the most conclusive. This is looking initially, Prodopedin was thought to be a dopamine modulator, particularly having impact on D2, D3, and this is using falopride, a D2, D3 receptor ligand, and you give falopride and you see very nice distribution throughout the basal ganglia, deep in the brain, and then you pre-treat with Prodopedin, and depending on Prodopedin's occupancy, you would then get elimination or diminution of that signal. What you see here is that there's no diminution of the signal, and that suggests that Prodopedin really at these doses is not binding and not preventing binding to D2, D3. This was very different with fluoxetine, which is a sigma-1 receptor ligand. And here, when you use fluoxetine, you see very nice distribution throughout the brain. You pre-treat with Prodopedin, and essentially the signal is almost eliminated. And this suggests that Prodopedin target is the sigma-1 and highly affinity and highly potent against that target. The sigma-1 receptor has been around for some time. It's a protein highly expressed in the brain. We know it has big impact on response to stress, and activation of sigma-1 has been shown in different models to have neuroprotective effects, improving neuronal viability, enhancing autophagy, increased connectivity, and decreasing stress. We've gone ahead to look at Prodopedin and its activity relative in vitro and in vivo models providing protection. This particular drug, Prodopedin, enhances autophagy, improves mitochondrial stress, decreases cellular stress, secretes, enhances in a dose-dependent way the secretion of BDNF, improves calcium homeostasis, improved spines, and decreased inflammation. So this was all over some years. This work was done by numerous collaborators around the world. This is work, for example, from Chris Ross's lab, where you can see cell death taken from both wild type and mutant animals. Here's the cell death with wild type. What you can see, the cell death with mutant is about 2.5 times that seen in wild type. And what you can see with up to 1 micromolar predopidine, you get complete restoration of neuronal viability. Furthermore, when you cross these animals to the sigma-1 receptor knockout, you lose the effect completely. The neuroprotective effects, and this is shown in every model, in all in vitro and in vivo studies, that the effect, the mechanistic effect, is completely mediated via the sigma-1 receptor. Now, for patients, and we hear and have heard this for so many years from patients from all over the world, and that is that the burden of HD leaves patients unable to perform daily activities, unable to function, and patients become dependent on others. And the TFC scale is a regulatory approved scale that measures the capacity to continue working, household activities, finances, and doing things that matter, eating, dressing, and taking care of themselves. And so TFC is a very important measure. It has widespread regulatory acceptance and endorsed by FDA and EMA as a primary outcome measure. It's been deployed for up to four decades. And the EMA, in our interactions with them, acknowledged the meaningfulness and acceptability of this as a single primary endpoint. FDA was of the same perspective that this would be a single primary endpoint. And so many have argued that any beneficial effect in TFC is clinically meaningful if it's significantly different to placebo. Now, in terms of placebo, why TFC is also helpful is there's no or minimal placebo response. When you look at TFC in a whole bunch of studies, what you can see, it goes down in early HD around 0.8 to 1 a year. And this has been seen before. In our prior PRIDE study, what we saw, and those were placebo studies, but here, just looking at drugs, there's been no drug thus far that has shown any impact on total functional capacity in terms of maintenance of functional capacity. In our prior study in PRIDE HD, we saw maintenance. This was not a primary endpoint. This was pre-specified for whole group, but also post-hoc looked at early HD. And what you can see is that in PRIDE, we already have some early evidence that this may be leading to maintenance of total functional capacity. We've gone on post-hoc to look for other evidence, neurofilament levels. And neurofilament is regarded as a measure of ongoing degeneration with reductions or even maintenance associated with clinical effectiveness of treatment. And certainly it appears that this is a measure of axonal injury. And so it's secreted from neurons following axonal injury. It increases, and Edward's lab and Lauren Byrne have shown beautifully how this increases with disease progression, and plasma NFL correlates with decline of cognition. And plasma NFL is decreased on treatment in other neurodegenerative disease, particularly seen in some trials in multiple sclerosis. And this is data from Lauren's paper just showing how in Huntington disease, there's an increase over time, over progression of illness. This is controls. These are early pre-manifest patients. And as the disease progresses, NFL increases associated with disease progression. Now, plasma NFL was not a pre-specified endpoint in PRIDE, but we did have plasma available from patients who had essentially pharmacokinetic PK studies. And we went back to look at all patients we could in terms of looking at placebo to see if there was any hint, any potential further support for believing that predopidine may be having impact. This was the change in TFC and NFL in the placebo group. And what you can see here, there's a correlation between change in TFC and NFL. As your TFC goes down, NFL goes up. And that's what we would expect. When we look at the trial itself, and this is in early Huntington patients, what you see is that in the placebo group, again, you get an increase in NFL. And this is very similar to that seen in other observational studies like in TRACK-HD and other studies. What we saw in our treated group in early HD was maintenance of neurofilament, or maybe even a small decrease. These were the actual data. These were not significant, but still showed in this small numbers of patients, the change in NFL essentially in the treated group was maintained, or small decrease went up. And when you looked at those patients for whom we had plasma and you looked at their TFC change, here you saw a big change. The 45 milligrams showed a significant increase, actually an improvement, and placebo showed the decrease that we would expect in a placebo trial for early HD. Now, there's a long, the other important finding is that this is a very safe and tolerable drug. It's been given in 1,300 patients in Huntington disease, 45 milligram. It's been given for a long period of time, 1,000 patient years. There's long-term safety data. And at this stage, the data suggests that the side effect profile is comparable to placebo. This is the compilation from numerous studies from all patients on 45 milligram BID. And what you can see, certainly the most common side effects, there's no increase seen in patients on predopidine. So this trial has leaders, PIs in North America is Andrew Fagan, Sandra Kostick is the co-PI, Ralph Riemann is the PI in Europe, and supported by Ann Rosser. And the study has been formally endorsed by EHDN and also it's been supported by the HDSA, HSC, and the EHA in providing support for the study. The study is phase 3. The first patient was randomized in October 2020. The primary endpoint is the change from baseline in TFC. It's designed to replicate prior findings that we've seen. It's a 2-arm study. This is the optimal dose for seeing a therapeutic response, both accepted by all regulatory agencies, duration 65 weeks, and our sample size, giving us appropriate power, is 480 patients. Let me give you a summary of where we are with the study. There are 60 sites in 9 countries in Europe, 9 countries in Europe, 30 sites in Europe, and 30 sites in the United States, and all sites have been activated. If we look at the status as we are today, is that if you look at those patients who are screened or reserved for screen, essentially we're fully recruited, and we recruited ahead of schedule. Close to 400 patients have been randomized, and we're on target to complete our randomization by October, and this would be significantly ahead of schedule. We're also delighted to say there's been very few side effects, a low dropout rate, and also a low screen failure rate that further talks to the tolerability of this particular drug. Now, at the same time, the drug is also in a phase 2-3 trial for ALS. And the target validation in ALS comes from the fact that mutations in the sigma-1 receptor actually cause ALS. And furthermore, there's a gene dose response. These are patients who have complete loss of function mutations, frame shifts. These patients end up with 0 sigma-1. They have a juvenile form of ALS. These are patients with a missense mutation, with a functionally defective protein, but some activity in sigma-1, and they have adult onset ALS. And this was part of the first platform trial. This is funded by Mass General and the Healy Center. This was a worldwide competition. There were 4 drugs chosen for the first platform trial in ALS with numerous other companies, UCB, Biohaven, and others, and predopidine. And this is a platform trial in that placebo is shared across 4 arms. And so you're recruiting essentially each study will recruit 120 patients, 40 each arm for placebo, 120 to 160. And this study too is on track. Most sites have been activated. And this is also ahead of target to complete randomization by November 2021. Merit Kutkovitz is the principal investigator of this particular study. So with these studies ongoing, we expect to have a readout from the phase 3 study for proof by early 2023. assuming that randomization goes as well as we've seen, and the ALS study will be towards the end of 22. We're very grateful to the whole community that has supported this so profoundly, patients and families, the investigators, the principal investigators, the lay organizations, and the community that has continued to support this study, and families and patients who have enrolled and continue to play an important role in this particular study. Thank you all.
Thank you very much, Michael, for this presentation, and thank you very much to all the speakers for presenting the current status of their studies and approaches for Huntington's disease treatments. I think it was a very dynamic session. We are a little late, as said before, so let's stop right now for 5 minutes. We'll start again with the panel discussion in 5 minutes sharp. So please, all the panelists stay on board. And please, if you still have questions, do submit them through the Q&A. See you in 5 minutes.